So today what I want to talk to you about is enzyme regulation. So you already know what an enzyme is, and you know the basic terminology. You know how it lowers activation energy for a chemical reaction that would occur naturally but at a slow rate. So today I want to talk about how cells might regulate enzymes and their ability to carry out a reaction, and how enzymes are organized into pathways within the body. So let's get started. Enzymes, as you remember, are proteins. So they're present in the cell, is controlled at a very basic level by a process we call gene regulation or gene expression, which is the control of transcription and translation. But the cells also control the activity of enzymes that are already present by inhibiting them and by activating them with other molecules. So that's a pretty clever trick. Let's see how that's done. Here in the top frame in this diagram, you can see that's just a standard enzymatic reaction. Here's the enzyme. The enzyme has an active site. The substrate fits into it. The enzyme substrate complex forms, and then the enzyme does its magic and the products are formed. Imagine a different scenario in which there's a molecule that can fit into the active site, but itself cannot be converted into a product. And I imagine that this is a different kind of molecule. It's not the same molecule as the substrate. If the cell were to release such a molecule, the new molecule would compete for the active site with the substrate, and therefore it would reduce the rate of the enzymatic reaction. So this is a pretty clever way to regulate how fast an enzymatic reaction happens. And such molecules that are released to do this are called competitive inhibitors. Let's think of another scenario in which inhibition might be brought about. So if you look in the lower right-hand side of this diagram now, over here, you can see that we've imagined a molecule that can bind with the enzyme, not at the active site, but at another site. Here it is, down here. And in so doing, it changes the shape of the active site, or it prevents the enzyme substrate complex from making products. So either of two things could happen. It could either change the active site shape, or it could just prevent further action by the enzyme on the substrate. Shape of the active site is key to allowing an enzyme to function. So if you were to change the shape of the active site, this would either reduce the rate of the reaction or bring it to a complete halt. Such molecules are called non-competitive inhibitors, and once again, a cell could release these non-competitive inhibitors and control an enzyme reaction. In other words, decrease the rate of an enzyme-mediated reaction. What about activation? If you think about it, the non-competitive inhibitor suggests a way to increase the rate of a reaction. And if you want, you can stop the videotape right now and think for a second about what you think it might be. Okay, are you back? Well, suppose there was a molecule that could bind to the enzyme and instead of changing the active site in a bad way, actually improved the shape of the active site. In other words, such a molecule would activate the enzyme because it would make the active site more receptive to the substrate. Here, that's called an allosteric activator, and you don't need to know that word allosteric, but here we're fine calling it an activator. And cells use this kind of control frequently. There are many other ways that cells inhibit and activate enzymes, but those await a more advanced course, or you could try to think about what some of them might be. I want to talk a little bit about the rate of enzymatic reactions from another point of view, and that's from the substrate point of view, because the rate of an enzymatic reaction depends on the substrate concentration. So once again, now we're dealing with concentrations. Imagine a situation in which there's little substrate available, but a fixed amount of enzymes. Well, it would be logical to say that, little product is going to be formed in this case. The rate of reaction would be low because the substrate concentration limits the rate of reaction. But what if the concentration of the substrate was now doubled? Then it would be logical to suppose that the rate of reaction would double because most of the enzymes are just hanging out waiting for more substrate to come along. And now with more substrate, more of them can be put to work and they get more product made. Continued doubling of the concentration of substrate will continue to increase the rate of reaction. But at some point, the amount of enzyme would start to limit the rate of reaction because most of the enzymes would be occupied with substrate And so a doubling of concentration would no longer cause a doubling of reaction rate because most of the enzymes are busy with substrate, creating product. And then at some point, there must be some concentration of substrate above which the rate of reaction can increase no further. And don't forget, this whole scenario has been played out with the assumption up here of a fixed amount of enzyme. Let's think about that. We have for various concentrations a reaction rate that we've measured. When we talk about the reaction rate, we actually mean the initial reaction rate because, as you've probably figured out, the enzyme, by constantly using the substrate, is decreasing its concentration. So we really are talking about the initial reaction rate. But if we plot the initial reaction rate versus substrate concentration, we're going to get a graph like this purple line here, where at first there's a linear reaction, but then as more and more substrate is put in, all the enzymes are being occupied, and ultimately there's a maximal rate for that concentration of enzyme. Make sure you understand why the curve looks like this. That would be very important for you to figure out. And I'll also leave it as an extra credit question, which you can submit to me via email, to figure out why the curves of reaction rate look like they do for the competitive inhibitor in green and the non-competitive inhibitor in blue. So if you can figure that out, shoot me an email, you get a little extra credit. Finally, I want to talk about metabolism. Metabolism is this weird word we've been talking about all year, and metabolism simply means the chemical processes within a cell or an organism that maintain life. In the grand scheme, metabolism consists of all the chemical reactions that allow organisms to gain usable energy and then use that energy to build the molecules required by the cell in order to keep living. Therefore, we can think of two types of processes: catabolic processes, in which large molecules are broken down into smaller components, often waste molecules, and energy is obtained, and anabolic processes, by which smaller molecules are put together to form larger molecules needed by the cell to maintain life. So, for example, replication of DNA, is that catabolic or anabolic? Well, it's anabolic because nucleotides are chained together into a new large piece of DNA. What about the destruction of an old messenger RNA by those recycler enzymes? Well, that would be catabolic. You are taking apart the old RNA. Now, we can think of this overview of metabolism like this. Here on the upper part, we have energy delivering nutrients, and this is kind of assuming a heterotroph here. And then through catabolism, we get the low energy waste products, but we also get the buildup of ATP and these other molecules, which we'll learn about soon enough. And then the energy that is stored in them is used in anabolic processes to take building block molecules, which are called precursors here, and turn them into macromolecules. Finally, let's connect this back to enzymes. And the important thing about this is the chemistry of life is organized into pathways. And the pathways are a series of chemical reactions that are carried out by different enzymes. Each of these enzymes has to be transcribed and translated and then regulated by the various mechanisms. A metabolic pathway begins with a specific molecule and ends with a specific molecule, which we call a product. Each step in the metabolic pathway is catalyzed by a specific enzyme. In the next few days, we're going to learn 3 distinct metabolic pathways that are part of cellular respiration that are called glycolysis, the Krebs cycle, and the electron transport chain. So that's going to be our work in the next classes. Here, I will leave you with this picture of a metabolic pathway where we start with molecule A and then enzyme 1 causes reaction 1 to happen and we get molecule B, but that quickly feeds into the active site of enzyme 2, reaction 2 happens and makes molecule C, which immediately is picked up by the active site of enzyme 3, reaction 3 happens, and we get the product D here. This is what I mean by a metabolic pathway, and as I say, we're going to start to study them. So I hope today what you have learned is how enzymes can be regulated, all the way from the regulation of transcription and translation to the individual molecular level regulation of already produced enzymes that can both inhibit and activate an enzyme. I hope you can also have a good understanding of how the rate of reaction is dependent on substrate concentration, and you might suppose that a cell could control the amount of substrate that an enzyme would have to keep an enzymatic reaction low. And finally, I hope you understand the concept of metabolism and that our enzymes are organized into these metabolic pathways that carry out both catabolism, where large molecules are broken down into building block molecules and energy and waste molecules, and then some of that energy gets stored in other molecules, and then those energy storing molecules are used to do anabolism, where building block molecules are converted into larger macromolecules, and they maintain and reproduce the structures of life. That's it for today, and I'll see you next time.